Dear students, today we learn about Transdermal drug delivery system. What is Transdermal drug delivery systems? It is also known as a TDDS. These systems lying under the category of controlled drug delivery system. That means TDDS is a part of controlled drug delivery system. TDDS are adhesive drug containing device of defined surface area that deliver a predetermined amount of drug to the surface of skin at a program rate to reach the systemic circulation. That means TDDS are an adhesive drug containing device. Adhesive means the transdermal patch is adhered to the surface of the skin, that having a specific surface area. From that, amount of drug is delivered to the skin at a particular rate, and from this passing to the skin, it reach to the systemic circulation. Let's see the examples of transdermal products. So you can understand that why the transdermal drug products are developed. It is developed for the hormone replacement. The drug used is testosterone. For angina, nitroglycerin drug is used. For pain relief, fentanyl type of drug is used. Hypertension, clonidine. Motion sickness, scopolamine. Smoking cessation, nicotine drug is used. And for depression, selegiline type of drug is already available in the transdermal type of drug delivery systems. Now see the potential benefits, advantages of transdermal drug delivery systems. Therapeutic agent means the drug delivered at a controlled rate through skin into systemic circulation. Already we have shown that it is a controlled drug delivery systems. So the drug is released or delivered at a controlled rate into systemic circulation. Maintain plasma level of drug from 1 to 7 days. As you know that tablets are used for 1 day, you require 1 tablets, or for 1 day you require 3 tablets or more. But in case of transdermal drug delivery systems, you can maintain the plasma level from 1 day to 7 days also. Controlled delivery resulting in more reliable and predictable blood levels. It is a controlled drug delivery systems, so you can achieve the reliable and predictable blood levels. Improved absolute bioavailability due to avoidance of first pass hepatic and gastrointestinal metabolism. The major advantage of transdermal drug delivery system is it avoids the first pass metabolism, as drug molecules passes through the skin layers, so it doesn't go first pass metabolism, so that it improves the absolute bioavailability. For example, diclofenac, the drug, you know that it having the 50% metabolism of drug, so that if you take 100 mg of diclofenac, then only 50 mg of drug will give the effect. So in that type of drugs, transdermal drug delivery systems is most advantageous. Also, it is more convenient and improve patient compliance, because in case of intravenous injection, we are not able to take by ourselves, but in case of transdermal drug delivery systems, we can apply the patch to the skin surface very easily, and we can also remove whenever it is not required. So it is more patient compliance, increasing efficacy and decreasing toxicity level of drug when compared to oral dosage forms. So it indicates that there are efficacy of the drug is increased and the toxicity level of the drug is decreased by using the transdermal drug delivery systems. Limitation of transdermal drug delivery systems. The drug must have some desirable physical chemical properties so that they can easily penetrate through the stratum corneum. As you know that the skin is having so many layers. Generally it having the 3 layers. So the drug molecules have to pass these layers and goes into the systemic circulation. So the physical and chemical properties of the drug is very much important. We will see its what are the requirements for the selection of drug later on. And another important thing is the amount of the drug is very much important. It shows that not more than 20 milligram per day the drug dose therapeutic value is required for transdermal drug delivery systems. Otherwise, it is difficult to incorporate into the transdermal patches. Another limitation is skin irritation or contact dermatitis. Due to the drug molecule, excipients, permeation enhancers of the drug to increase the absorption is another limitation. As you know that the transdermal patch is applied to the skin surface, and the transdermal patch contains so many things. One is drug molecules, excipients and permeation enhancers for the drug. So that, and also you know that the skin barrier is also, skin surface is also different from person to person, age to age and so many things. So sometimes these drug, excipients, permeation enhancers creates skin irritation to the surface of the skin. So it is another limitation. Clinical need is another area that has to be examined carefully before a decision is made to develop a transdermal product. That means we cannot take any of the molecule for the development of transdermal drug delivery systems. First of all, we have to identify what is the requirement, why transdermal drug delivery systems is required to be developed. And on that basis, we can develop the transdermal drug delivery systems. Another limitation is barrier function of the skin. It changes from one side to another side on the same person, from person to person and with the age. So it is also one of the limitation of the skin, and that also creates amount of drug permeated through the skin is also different. Here we see the structure of human skin. Already you have learned into the human anatomy of the skin, our body. Basically there are 3 layers: epidermis, dermis and subcutaneous tissue. From this structure of the skin, this duct of sweat gland, hair follicles are also important for the transfer of drug to the systemic circulation. This is the pathway of drug transport across the skin. Basically, there are 3 pathways, 1, 2 and 3 for the transfer of drug, penetration of the drug through the skin. First one is through the sweat ducts, the drug molecules can transfer into systemic circulation directly across the stratum corneum, and third one is through the hair follicles. Here we see the basic components of transdermal drug delivery systems. The requirements for the development of transdermal systems. First one is polymer matrix. Any of the controlled drug delivery systems are not prepared without the polymers. because the polymer controls the release of the drug from the device. Here some of the criteria are mentioned which are important for the selection of the polymers. First one is molecular weight and chemical functionality of the polymer should be such that the specific drug diffuses properly and gets released through it. Because the selection of the polymer is poor chemical properties and molecular weight, then you cannot target the drug as per your requirement. So it is very much important. Another is the polymer should be stable, not affected by any of the like heat, moisture and other parameters. Non-reactive with the drug. If it is reacted with the drug, then it causes degradation of the drug molecules. So it should be not reactive, easily manufactured and fabricated into the desired product, and inexpensive. The polymer should be easily prepared, easily manufactured and easily available so that the cost of the polymer is less. The polymer and its degradation products must be non-toxic and non-antagonistic to the host. It should be non-toxic. The mechanical properties of the polymer should not deteriorate excessively when large amounts of active agents are incorporated into it. That means when you add the drug molecules with the polymers in large quantity, the mechanical properties of the polymer should not be changed. These are some examples of polymers. Polymers are of three types: natural polymers, synthetic polymers and semi-synthetic polymers. The natural polymers includes cellulose derivatives like HPMC, HPC, zein, gelatin, waxes, proteins, natural gums, natural rubber, starch, etc. Synthetic elastomers, that includes polybutadiene, polysiloxane, silicone rubber, butyl rubber, neoprene, styrene butadiene rubber, like that type of rubbers. They are having the adhesive properties, so they can be used. Synthetic polymers like polyvinyl alcohol, polyvinyl chloride, polyvinyl pyrrolidone, polypropylene, polyacrylate, polyurea, epoxy, polymethyl methacrylate. This type of polymers, they are synthetic in nature, they can be used. Another important basic component is drug molecule. These are some requirements, criteria for the selection of the drug for the preparation of transdermal drug delivery systems. One of that is potency of the drug. It should be less than 200 mg. If you can select high amount of drug for the TDDS, then it is difficult to incorporate drug molecule into the TDDS. For example, the drug like paracetamol, the dose is 500 mg. So it is very much difficult to include 500 mg of PCM into the transdermal drug delivery systems. So the drugs like fentanyl, clonidine, diclofenac, those are having less amount of drug. They are incorporated into the transdermal drug delivery systems. Lipophilicity, log P value should be in the range of 1 to 3. Molecular size less than 500 Daltons. If it is more than that, then it is again difficult to incorporate. Irritation should not create any irritation to the skin. Melting point less than 200 degrees centigrade. Immunogenicity should not stimulate any immune reaction to the skin. Other important considerations are pharmacokinetics. half-life should be short.